In our previous videos, we have discussed about 2 marks of questions and 3 marks of questions. In this third revision video, we are going to discuss about questions which is related to 4 marks. Right? Now here, according to our blueprint, that the 35 number of question, it will be from second chapter, and the alternative question will be also from the second one. In 36 number, it will be from chapter number 5. 37 number, it will be from chapter number 6, and the alternative question will be also from chapter number 6. 38 number, that will be from chapter number 10, and 39, that will be from chapter number 11. So let's start our journey with the 35th number of question, that will be from chapter number 2. Now, as we all know that in the discussion of 2 marks and 3 marks of the second chapter, right, in this session of 4 marks of question from second chapter, we will have some questions. If we start the discussion, so we will start our discussion from the chlor-alkali method. Okay, chlor-alkali method. So first of all, why this method is called chlor-alkali method? Because during this, chlorine and alkali means base, both are produced. So that is called chlor-alkali method. Now let's understand that what is chlor-alkali method. Brine, so that's the saturated aqueous solution of salt. Means we can understand it like this: NaCl plus H2O. NaCl plus H2O means the brine solution. Right? Here the brine solution is there. Here we are having the membrane, and anode and cathode is placed. As we all know that anode means positively charged and cathode means negatively charged. So this is our anode and this is our cathode. Okay dear. Now see, when NaCl plus water is here, between this anode and cathode, there will be the electrical potential. So when electric current will pass from this type of electrolytic cell, so what will happen? There will be splitting of sodium positive ion, chlorine negative ion, hydrogen positive ion, hydroxide negative ion. As beta you can see that this both negative ions, chlorine and OH, that will move towards anode. And sodium positive ion and hydrogen positive ion, that will move towards negative electrode, that is our cathode. Now during this reaction, what we will get? We will get NaOH plus hydrogen gas is released and chlorine gas is also released. This will be in aqueous form, water will be in liquid form, and brine, that's in aqueous form. To balance the chemical equation, we have to write here 2. Now we can say sodium is 2, so sodium is 2, chlorine is 2, here also the chlorine is 2. Now hydrogen is 2, hydrogen is 2, and oxygen, here we are having 1 oxygen, here we are having 2 oxygen. So what we can do? Okay, hydrogen is 4 total, so we can write here 2. Now our equation is balanced. Now see beta, this hydrogen, it is positively charged, as we know that H plus ions. So where it will go? It will go to the negative one. So it will be deposited on cathode. Here you can see that the hydrogen is removed from the cathode side, and the chlorine Cl minus, that is negative, so it will go towards the positive electrode, so it will collect on anode. As you can see here, the oxygen is collected. So you can see that during this process, alkaline means base, NaOH, and chlorine is formed. So that's why it is called chlor-alkali method. So as we can see here, the hydrogen that will be used in fuel, in production of ammonia, and chlorine, here there will be chlorine. So this chlorine gas, it will be used in PVC pipes, right, as a monomer of PVC pipe. Also it will be used in water treatments, okay, and also in bleaching of water, means making the water germ free. In swimming pools also this chlorine is used. So this way we can understand chlor-alkali method. So this is the important question that it can be asked to you in exam, that write short note on chlor-alkali method, or write the production of sodium hydroxide, okay, or the electrolysis of brine. So if in any of this question is this type of questions, you have to write chlor-alkali method. So keep that in your mind, how we are going to perform the chlor-alkali method. First we will draw the figure, then we will write its description, after that the equation, and then we have to also write the uses of chlorine and uses of hydrogen. Now if we talk about the another question from the second chapter which can be asked for 4 marks, that is the production of hydrogen gas, right? Already we have done this experiment, okay? So what will be there? Simple. We will take one test tube, in that there will be zinc granules, and we are adding dilute sulfuric acid or dilute hydrochloric acid, any of acid you can use. So there will be evolution of gas. As you can see that this hydrogen gas, it will get evolved, and this hydrogen gas, it is collected by downward displacement of water. Now beta, see, if we want to write its chemical equation, how we can write? Zn plus HCl gives ZnCl2 plus H2. This will be in solid form, this is in aqueous form, this will be also as aqueous, and this will evolve as gas. For the given one, what we can say? Zn plus H2SO4, that's an aqueous one. It gives ZnSO4, that's aqueous, plus hydrogen as a gas. Okay, so this way also we can write its equation. So this question can be also asked to you, that explain the activity in which acids react with metal to give dihydrogen gas, or the production of dihydrogen gas. It can be also asked like this, that write about production of hydrogen gas in laboratory. So in that question we have to do this experiment, we have to write this experiment. The hydrogen gas which we are getting from the downward displacement, what we can say? We can say that it is lighter than the air, it is insoluble in water, right? It is colorless, and if we take out the candle near that gas, so it will be combustible. Okay, so this are the properties of hydrogen gas which is produced during this type of reaction. So beta, we can understand the chlor-alkali method and the production of hydrogen gas in laboratory as a 4 marks of question. There are also other questions which can be asked to us in 4 marks from chapter number. Other questions which we can discuss from chapter number 2 for 4 marks, that is preparation and uses of baking soda and washing soda. Here we will discuss about preparations of baking soda and washing soda, because uses are easy, you can prepare it by yourself, right? For baking soda, what we have to do, right? First of all, we know what we can do for baking soda. We want normal salt NaCl plus carbon dioxide plus water and plus ammonia. What it will give us? It will give us NH4Cl plus NaHCO3. This NaHCO3, which is also known as sodium hydrogen carbonate or sodium bicarbonate, this is our baking soda. We can also call it as sodium bicarbonate. That's our baking soda. When beta we heat this baking soda, so it will give us Na2CO3 plus H2O plus carbon dioxide. Okay, now so see beta, this is our baking soda, and uses of baking soda we know, which makes the pakoras, crispies and all. Okay, so this way the preparation and uses both can be asked to you in 4 marks. We can also think about washing soda. Washing soda means that is our sodium carbonate. Now how the sodium carbonate is prepared? So Na2CO3 into 10H2O, sodium carbonate decahydrate. Okay, so that will be our washing soda. When we heat this, there will be decrystallization of water like this, Na2CO3 plus 10H2O. Okay, so this way we can also understand the washing soda. How we can obtain the washing soda? Here is the another one, that if we are having baking soda and just we heat it, so we can get sodium carbonate, right? And if we do crystallization of it, so that will be result into washing soda. Uses of washing soda is also very easy. That also we can prepare by ourselves, so we are not discussing over it right now. About bleaching powder, so CaOH twice, slake lime, and when chlorination of slake lime is done, we will get CaOCl2. What we can get? We can get CaOCl2 plus H2O. This CaOCl2, that is known as our bleaching powder. Where this bleaching powder is used, as we all know that in textile industry to bleach the clothes, also it is used to make water germ free, also as a agent, as a reactant. It is used in different industries. So this way with our different preparations and their uses can be asked to you in 4 marks from our second chapter. Also there is one experiment that it is what happens when acid it reacts with metals carbonate and metals bicarbonate. So reaction of acid with metal carbonate and with metal bicarbonate, that is also the important question for 4 marks. In terms of experiment it can be asked to you. So hope you know that, and that I am going to give you as a self study. You can prepare it well. Now let's talk about 36th number question, that will be from chapter number 5. As we have discussed that the questions of 2 marks, 3 marks or 4 marks, it is dependent on the paper setter, how the paper is set. Means it can take the whole question of 4 marks also, or it can divide the question. Means it can ask A and B. So each will be of 2 marks, otherwise A is for 1 marks and B is for 3 marks. That way also the question can be asked. If we talk about the 5th chapter, so there will be this type of, it's a probability that most of the question from chapter number 5, if it is in 4 marks, so that will be not a whole question. There will be different sections of it. If it is like A and B, so it can be possible that question A is of 2 marks and question B is also of 2 marks. Otherwise A is of 1 marks and B is of 3 marks. Okay, so it's necessary to prepare all of the short topics also for our 5th chapter. Now let's discuss about some of that. In 3 marks of topic we have discussed about triads of Doberer. So it can be also like this, that one question is there that is of 1 marks, and in the another one they can ask you triads of Doberer. So that pattern can be also followed for 4 marks for our 5th chapter. Now with some remaining points, let's discuss that of chapter number 5. So in that we can think about limitations of Newland. So what was limitation of Newland? So Newland law was only applicable till calcium, means till 1 to 20 element it was only followed. Second one, at that time only 56 element was discovered. So Newland has applied that law for only 1 to 56 elements. Third limitations of Newland was that when the arrangement was done, right, so in some of his group he has placed the element which was not having similar property to the other one. So we can also talk about that limitations of Newland, which can be asked to you in 2 marks or 3 marks. Okay, also we can talk about limitations of Mendeleev's periodic table. Right, so as we have discussed that in 3 marks of question also, but again it can be also asked to you in 4 marks. So what is limitations of Mendeleev's periodic table? So first limitation, that's position of hydrogen. We have discussed about it. Position of hydrogen. Sometimes hydrogen behave as metal and sometimes it behave as non-metal. So where to place that hydrogen, that was the question. It was not explained by the Mendeleev. It's given there in the notes. You can prepare very well from the notes and from whatever you have written in your notebook. Second one, that is position of isotopes. First of all, we must know what is called isotopes. Isotopes means same atomic number and you will have different atomic mass, like 1H1, 1H2 or 1H3, protium, deuterium and tritium. These are isotopes of hydrogen. Now, as we all know beta, that the Mendeleev's periodic table, it was related to atomic mass. Here, for each of the isotope, their mass are different, but the element is same. So, due to this, where to keep this element, it was the question mark, means it was not explained by law of Mendeleev for its periodic table. So, that will be also our limitation for Mendeleev's periodic table. The another one which we can say, that there was irregularity in the atomic mass, in the increasing of atomic mass, there was an irregularity. So that was also the limitation of it. And the fourth one, as we know, that some element which were lighter, right? Okay, and which some element which was heavier, it was kept before the lighter element. Okay, so that was also the limitation. So limitations of Mendeleev, it's very most important topic for 3 marks also and for 4 marks also. Now, some of the 1 marks of question which can be asked with this type of 3 marks of question is, what is modern law of periodicity? So what is modern law of periodicity which we are following in modern periodic table? Yes, that's right, that all of the properties and functions, right, all of the properties of element, it is dependent on their atomic number, right? So that is our modern periodic law. And what is the periodic law of Mendeleev? That all of the chemical properties and its function, it is dependent on its atomic mass, right? So that periodicity for Mendeleev's periodic table and also for modern periodic table, we have to understand, which can be asked to us. Okay, so this way the periodic law we can understand for Mendeleev's periodic table and for modern periodic table. For Mendeleev's periodic table, what it is, that it is the function of their atomic mass, right? And for modern periodic table, it is function of their atomic numbers, right? So in Mendeleev's periodic table, it was based on atomic mass, and in modern periodic table, it was based in atomic numbers. Now, as we all know about modern periodic table, that it is classified into 18 groups and 7 periods. Okay, now groups means what and periods means what? So we can understand that groups is vertical lines and periods is horizontal lines in our modern periodic table. Now, if we talk about some trends, right, related to our modern periodic table, here we are going to discuss the 2 trends of it. First is atomic radius, or we can also call that atomic size. More radius means more in volume, more in size, right? So that way also we can understand the atomic radius. So as beta we have studied that when we are moving from up to down, means when there is increase of atomic number in the group, when there is increase of atomic number in the group, electron is added into the new cell. So as we move from up to down, so what will happen to atomic radius? In group, if we increase in group, atomic radius is increased. So what will happen in periods? Okay, so as we move from up to down in for atomic radius, the new electron enters into the new cell, so the atomic radius will get increases. So as we move from left to right, so it will get decrease. So in periods, by increasing it, it will get decrease. Okay, so this way we can understand. It is clear that whatever change is done in group, the opposite change you will see in period. Means in group, when we are moving from up to down in group, if it is increasing, so when we will move from left to right in period, it is going to get decrease. Okay, we can also talk about the metallic properties. So beta, we also know that according to the metallic properties, we are having metals, semi-metals or metalloids, and we are also having non-metals. Now, as we know that as we move from left to right, here are non-metals and generally here are metals. So when we move from left to right, metallic properties in periods, it is decreasing. So when we move from up to down, right, so the electron will enter into the last orbit, and as it is far, it's easy to remove the electron, so the metallic properties is going to get increased. Okay, so from the topics of trade, it can be asked to you as a 2 marks, and that way A and B, it can be asked that 4 marks of one question, but it can be 2 marks of 2 questions also for 4 marks from our chapter number 5 for 36 number of. Now let's talk about 37 number of question, that will be from our 6th chapter. As we know that in 6th chapter there are many questions which can be asked to us in 4 marks. So let's start our journey from the respiration. Okay, so beta, what is respiration process? So the food which we eat, it is digested and it is converted into absorbable components. Now that absorbable components, it will get oxidized and it will release energy. So that process is called a respiration process. We know that there will be 2 types of respiration: aerobic respiration and anaerobic respiration. Aerobic respiration means what? Which is done in presence of oxygen, and anaerobic respiration means it is done in absence of oxygen. We have also understood the difference between aerobic respiration and anaerobic respiration in our previous videos, and you have also prepared the note and you all know about it. So we are not going to discuss it in detail right now, but we can discuss about breakdown of glucose molecule. Okay, so that can be also the important topic for 4 marks or 3 marks for our exam. So as we can see that glucose, it is 6 carbon containing compound. So in cytoplasm, right, this glucose will get converted into pyruvate, which is 3 carbon containing compound. Now here we have discussed the 3 cases. That first is in absence of oxygen, second is the lack of oxygen, means less amount of oxygen, and third is in presence of oxygen. So let's start. If there is absence of oxygen and this pyruvate is going to get breakdown, it is going to get decomposed, so it will be done in the medium of yeast. So that is our plant medium, as we know. So in that case, what we will get as a product, we will get ethanol, which is 2 carbon containing C2H5OH plus carbon dioxide and energy, but it is in absence of oxygen, so not proper breakdown of pyruvate will be done, so this energy will be very less. The second case is that if it is done in lack of oxygen, means not sufficient amount of oxygen is there, so that can be done in our muscle cells, and as a product what we will get the lactic acid, which is 3 carbon containing acid, and the energy will be released. But this energy will be also in less amount because there was lack of oxygen, so not a proper breakdown is done. Now the aerobic type of respiration, in that the presence of oxygen, which is done in mitochondria. We all know that mitochondria is known as powerhouse for our cell, right? From that it will get converted, this pyruvate will get converted into carbon dioxide, water and energy. This energy will be in very more portion because this breakdown is done in presence of oxygen. So beta, we can understand this breakdown of glucose by this chart, and we are also able to write the short note on breakdown of glucose. Okay, so this way we can understand it. The another question which we can understand, that is digestion in amoeba. So how digestion in amoeba is done? So as we recall the process, first there will be ingestion. So amoeba will take the food inside the body with the help of pseudopodia, right, or false leg. We can also call it as false legs, right? It will take the food inside its body, so that is our ingestion process. Then it will create a vacuole, right? So first it will come to ingestion. After it is injected, there will be digestion, so it will get digested. After it is digested, that will be absorption, means the useful components it will get absorbed by the cell. After it is absorbed, so after that there will be assimilation, that is obtaining energy from the absorbable compounds, and at last we will have egestion, and by that egestion also the undigested food particles, it will remain, it will thrown out by the process of diffusion. So beta, we can understand it by ingestion digestion, absorption, assimilation, and digestion. Okay, so this way we can also understand the nutrition in. Now the important one, that digestion in human being. Okay, so as we know that digestion in human being, it starts from mouth. We enter food through mouth. In mouth there will be teeth. Okay, we can understand it by this mouth. In that there will be teeth, tongue, and salivary glands. This salivary glands, it secretes saliva. In that saliva there will be amylase enzyme which converts the starch of our food into maltose. Okay, after that the food will go to from esophagus or the food pipe. We can also call it as food pipe, or we can also call it as esophagus. It will go to stomach. In that stomach there will be secretion of HCl, pepsin, and mucus. Okay, so what is function of HCl? It kills bacteria, it gives us the acidic medium, and it breaks down the food into small globules. Pepsin, it is used in digestion of protein, and mucus, it protects our cell wall from the more secretion of HCl in stomach. Right? From that it will go to small intestine, then to large intestine. Now as you know that in small intestine absorption of that food will be done. Here also, like liver, pancreas, and the another one, that is our bile duct is there. From that the bile juice will secrete it. Bile juice is an alkaline medium, that's a basic medium, so it will neutralize the more acidity of the food. Pancreas, it will secrete the pancreatic juices, right? In that there will be amylase, trypsin and lipase. What is the function of amylase, trypsin and lipase? That also we know that amylase is used in digestion of carbohydrates. Trypsin, it's used in digestion of protein, and lipase, it is used in digestion of fatty acid. Okay, so that enzymes, what is their function? That also we can understand. Pancreatic juice. Okay, and in small intestine there will be finger-like projection, which is also known as villi. By this villi, the surface area will get increased and food will get absorbed, and after that it will move to our large intestine. Right, between that the regulation of food from stomach to small intestine, there will be a sphincter muscle. One more thing, why this is small intestine and large intestine? It is due to its width. But if we talk about the length, so small intestine is the longest part of our alimentary canal, but according to its width it is small, so that's why it's called small intestine. After that the food will move in large intestine. In large intestine the reabsorption of water and the nutrients is done, and by anus it is thrown out from our body. Okay, so we have just taken an overview of how digestion is done in human beings. If we talk about our respiratory system, so as we all know that nose, in that there will be nostrils. Okay, by these nostrils we are inhaling the oxygen inside our body. If we talk about the mechanism together, so we can talk about the diaphragms. Now beta, what happens when the diaphragm it moves in downward direction, so there will be chest cavity increase in our body. So what will happen? Oxygen will enter through our nose, through nostrils, and then mucus is there, so it will get purified. Okay, and it will go to our trachea, from trachea to bronchi, then to bronchioles, and then to alveoli. In alveoli the exchange of gas will take place, and the carbon dioxide is exchanged by the oxygen, and at that time our diaphragm will get contrast and it will move in upward direction, so chest cavity will get decreased and the carbon dioxide will be thrown out from the same path. Okay, so that how our respiratory system is, how it functions, and how diaphragm is responsible for our respiratory system, means the mechanism of our breathing, mechanism of our inhalation and exhalation we can discuss, right? And we can also write it in a particular method that this way our respiratory system is working and this is our mechanism. So we can divide these questions, right? The how our respiratory system is and what is mechanism of our respiratory system in 4 marks. This respiratory system of human is also very most important question for 4 marks for your board examinations also and for your unit test also. Okay beta, we have also discussed about our excretory system in 3 marks of topic. So that topic can be also asked to you in 4 marks, but a little bit will be added there. So not only the excretory system, also the urine formation can be asked to you. So in that case, you will explain our excretory system and you will explain the urine formation, right? As you know that from the Bowman's capsule, right, under the higher pressure, means that's known as ultrafiltration, the urine will pass right from the Henle's loop and it will go to ureter, to it will be stored in urinary bladder, and by the compression of urinary bladder it will be thrown out from our body. So this way the digestive system, respiratory system and excretory system, these topics are very most important for our exam and for board exam for 4 marks from our chapter number 6. Now let's discuss our chapter number 10 for question number 38. See, already we have discussed many of topics in 2 marks and 3 marks from our chapter number 10. So some of the important topics, right, which can be asked to you in 4 marks for from chapter number 10, right, that can be more probability the image formation. Here I have tried to explain some important image formations, and rest also you can do it by yourself. See how it can be asked that draw the figure and write the position of image and also the types of image and dimensions of image if the object is beyond 2C. So here we can say that the object is when beyond 2F. So when the object is beyond 2F, where we are getting the image? We are getting it between F2 and 2F2, right? Okay, the fundamental laws that we know that what it will be. So simple thing it is that first ray which is passing parallel to our principal axis, it will pass from F at other side. Second, that our optical center, it will not return, it will pass without any type of refraction. Okay, these 2 rays generally we are using. Third, also we can understand that which is passing from F, that will get parallel to our principal axis. Okay, so these 3 fundamental rays, by using these 3 rays, right, we are able to find where our image will form, where the 2 rays are intersecting each other. At that point we are getting our image. So how it can be asked that if the object is beyond 2F1, so where the image will be? It will be between F2 and 2F2. How it is? So as we can see that it is smaller in size, right? How it is? It is real but it is inverted. Okay, so this way the another one, that if the object is at 2F1, where we will get the image, we will get it at 2F2, same in size, right? So as we can see here, now on 2F, again it will be on 2F, that between 2F and F, so between 2F and F1, where it will be, it will go beyond 2F2, it will be enlarged, this will be enlarged. If it is on F, so that will be at infinite distance. If it is at infinite, then we are getting at F. So beta, all these image formations, this we have understood about convex lens, right? So we are having different image formations for concave mirror and convex mirror, and also we have it for convex lens and concave lens. Okay, so the image formations from that, any of 4 images can be asked to you in 4 marks for this question number 38, which will be from chapter number 10. Okay, also some other topics can be there like magnification into marks and power of lens in 2 marks. That way also the 4 marks of question can be there. Also the refraction by glass slab, it can be also there in 4 marks. Already we have discussed that questions for 3 marks. Okay, so this way, this image formations, we can think about the image formations, that 4 different image formations can be asked to us in 4 marks from our chapter number 10 in question number 38. And rest of the topics, important topics, we have discussed in our 2 marks and 3 marks of session. So just go through it also, because some of the points from that also can be included as a mixture of different questions in our 38 number question, which will be from chapter number 10. After discussing that, now we will go towards our last question, that is 39, and for that we will have a question from our chapter number 11. Now here 4 marks of question from chapter number 11, right? Already we have discussed many of questions in 2 marks and 3 marks, but the refraction due to prism, it can be asked to us in 4 marks. Now let's see how it is done. Suppose we are having a prism A, B, C. Okay, incident ray, whatever name you want to give it, PQ, here I am giving the name, M dash is our normal, right? Okay, this is our angle of incident I, incident ray and normal angle between them, that is our incident angle. Okay, here. Now here also we can draw the normal N dash, and it exists, right? So this is our emergent ray. Angle between that E, that is our ray of emergent. Now beta, if we extend this rays like this, and the angle here which we are getting, this angle is called our angle of deviation, delta, or it is known as D. This angle A, this is our prism angle. What it is? It is our prism angle. Okay, so what we can say beta, that angle I, that is angle of incidence, angle E, that is our angle of emergent, angle A, that's our prism angle, angle D or delta, that's our angle of deviation. Okay, the relation between this 4 angles, how it is, that is known as prism equation. That's angle A plus angle D is equal to angle I plus angle E. From that, how we can find out the angle of deviation? Angle of deviation D is equal to angle of incident plus angle of emergent minus our prism angle. Okay, so beta, this question, refraction through glass prism, can be asked to us, right? In that we have to draw the figure, we have to explain the figure, then we have to note these angles, and after that we can give the relation between these 4 different angles, and we can find out the angle of deviation. Okay, so if it is asked in 4 marks, you are going to draw the figure, you will explain the figure, right, and then the interrelationship between that angles. So this way refraction by glass prism can be asked to us in 4 marks of question. The another question which we can think from our unit number 11, right, chapter number 11, that is dispersion of light and recombination of light. It's very important question, right, for 4 marks. So beta, what will be there in dispersion? So as we all know that white light and the separation of white light into 7 different colors, V, I, B, G, Y, O, R, right? So white light, it is separated into 7 different colors. That phenomena is known as dispersion of light. How it will get dispersed, right? V, I, B, G, Y, O, R. Violet, indigo, B for blue, G for green, Y for yellow, O for orange, and R for red. Okay beta, one more thing which we know, that more velocity less deviation. So velocity is inversely proportional to deviation. Now see, as we know that violet color, it will have a less wavelength, so its velocity is also less. Less velocity means less wavelength means less velocity, and less velocity means more deviation. That's why violet color you are seeing more deviated from its original path. Now red color, it is having more wavelength, that means it will have more velocity, and as there is more velocity, there will be less deviation. So here you can see that there will be very less deviation angle of red color. Okay, so this way beta we can understand the dispersion of white light. So what is dispersion of white light? So the separation of white light into 7 different wavelengths, so that is considered as dispersion of light. From that we are getting the spectrum. From downward to upward, it will be violet, indigo, blue, green, yellow, orange and red. With this also it can ask that recombination of dispersed white light. So how we can do? We have to take 2 prisms of same prism angle, and it is arranged as shown in figure, right, opposite to each other. By this first, what will happen? The light, it will get dispersed, right, and by the second one, the dispersed light is going to get converged. So by our first prism, our prism A, that is going to disperse our light, and our prism B is going to recombine our light. Okay, and this type of experiment was first done by Sir Isaac Newton. Okay, so this question is also important, that write short note on dispersion of light and also explain the recombination of light. So we have to draw the figure, we have to write the definition of dispersion, then we have to perform the activity. After that we have to write the spectrum, their colors, and we have to explain the relation with the wavelength, velocity and the deviation. And in recombination we will also draw the figure, and we will explain how by first prism it is dispersed and how by second prism it is recombined. So beta, this way we have understood our 4 marks of questions. We have understood our 4 marks of questions from question number 35 to question number 39. Okay, in our revision video 1, in our revision video 2, and in our revision video 3, we have almost discussed the 2 marks, 3 marks and 4 marks of maximum questions. Again, it is not necessary to ask 3 marks of a single question. They can take 2 different questions and can make it of 3 marks, or in 4 marks also it is not necessary to ask only a single question. They can take A, B, C, D, or they can take A, B, or they can take A, B, C, and that way also they can ask us. Now for the 1 marks of question, if you have prepared this 2 marks, 3 marks and 4 marks perfectly, right, so I don't think so that there will be more any confusion, and I think you will not require any of thing to prepare for 1 marks, because almost it will be completed. And if you have done the thoroughly reading of our textbook, if you will go throughout our worksheets and our other revision videos, so you will be able to answer the 1 marks of question as well as 2 marks, 3 marks and 4 marks of question. So everything is just provided to you. You have done very well work, right? So from your notes, by watching the videos, by your dedication and by your hard and smart work, you will achieve your goal and you will score very more in your exam. So all the very best for it. Thank you.